In this opportunity, I want to present the optical and fluorescent microscope that we have in the laboratory. This microscope is a vertical instrument in which we can observe different samples. We use normally this microscope to observe biological samples prepared before we observe it in the confocal or in the AFN. Also, we try to use this microscope to verify that we have the samples, for example, cells or nanoparticles, in the media that we prepared before we use it in other microscope. This microscope have two different sources to observe, and the most advantage of this is that we can observe fluorescence because we have the filters and also a mercury lamp that can be used for that. This microscope have an halogen lamp, and as you can see, the start of the instrument is very simple. We turn on the microscope and then the LCD screen. The advantage of this LCD screen are that we can put scale bars immediately when we observe a sample. Also, we can draw or mark different parts of our sample. As you can see, in the instrument is very simple. We have binoculars, we have different filters that you can see in the body of the instrument, and also in the posterior part of the instrument you can see the halogen lamp that allow us to see different samples with different intensities that we can change normally with the source of light. Also, as you can see, we have different objectives from a low magnification, that is 4x, to allow higher magnification, that is 100x. As you can see in this part of the video, we can observe the Nikon mercury lamp. This Nikon mercury lamp have an advantage because we can observe the fluorescence of different samples. For instance, we use this mercury lamp to observe samples that can be marked or dyed with DAPI, FITC, and FTRAC. That are samples that can be achieve different fluorescences without use the confocal, that is a maximum equipment. Also, this mercury lamp have a counter in which we can observe the running time of the mercury lamp, because the problem with mercury lamp is they have a time for use. For this microscope we use the fluorescence before we use the confocal, so we are sure that we are preparing good our samples. As you can see in this part, we can use the different parts of the instrument to focus our sample in the white field and start to observe the sample in the LCD screen without use the binoculars. That are a very advantage of this microscope. Also in the LCD screen, as I say before, you can start to put different information and keep it in a USB without have to use another computer or something to keep your samples. In this screen also you can change the intensity of the light, you can change the parameters of the microscope, the parameters of the objectives, you can change the objectives, and also you can use the most important part, that is the scale bars, because we needed to measure our samples in case of these fibers or cells that are presented in different materials, in different biological materials. For instance, in this opportunity we use the light microscopy to check our samples. This is a first step to the other microscopes, and as you can see, you can move the part of the sample that you needed using the platform in which the sample is put it, and you can also be more zoom out or zoom in depending of you need. If you need to see more detailed parts, I show you this paper that was presented for Nicolas and Alvarez in 2019 from tomato root, and as you can see, the light microscopy allow us to see a structure similar to the structure in the SEM. Also, Vicente Flores et al. In 2020 presented this paper for light microscopy using Quajilote, that is. And now I want to introduce the atomic form microscope, or AFM. This microscope can be used in different fields of investigation, but particularly in all these fields in which force in nanonewtons or scanning in nanometers can be used. This microscope have different advantage in comparison with other microscopes, but essentially is a very simple optical mechanical instrument in which a holder of the sample that you can see in the center of the image can be used to scanning a sample topographically. The main parts of this holder sample are the holder of the cantilever. As you can see, this holder of the cantilever have the cantilever, that is the main part of the scanning, in which a tip, as you can see in the image, can scanning directly the sample. This tip is in the order of the nanometers and can be used for not only scanning and also for indentation. In this part of the instrument is the holder of the cantilever, in which we with these two different instruments we can put the sample to attach very good. For the initial part, we use the Nano Drive software. This Nano Drive software allows us to select which type of method we can use with the AFM. We have contact mode, tapping mode, and not contact mode. These different methods, as you can see, can also be used depending of the sample. For example, we have also tunneling, contact with SC, stem. That depends of the adjustment that the equipment have. In this case, we use contact mode to observe a sample that is the typical sample for calibration. After a few minutes, we start to introduce the parameters of the equipment that allows us to check our sample with very good characteristics. As you can see in this image, this is the main part of the software. We can observe in one screen the tips or the cantilever in which the tip is attached and the sample. For other hand, we start to check first the lasers because the laser have to be in the center of the photomultiplicator to obtain a very good and substantial information for the sample. If this is not, we use these controls, manual controls in the holder of the cantilever, which allow us to center the laser in the part that we needed. After we center the laser and we check that the laser has the most potential, as you can see in magenta, we can start to make a scanning of the sample. To scanning the sample, we have to select this tool that is called engagement, in which the sample allow us to scanning when the tip start to approach them. The tip start to slowly go down until they touch the sample, and after that time the software allow us to select what time of scanning we need, the image size. We can store the image in our folders. Also, we can change the full range of the scanning, but after start the scanning, we can observe what happened with the sample when the tip start to scanning. As you can see in this profile, we have clean line, because maybe in this part of the our sample we don't have anything with a topography, in which the tip, as you can see with the laser in red, start to touch the sample. In this image, it's very important to observe that we can have different tips in one cantilever, but only with attaching the laser in one of them we observe a very good image. After one time, we start to observe changes in the profile because of the topography of the sample. As you can see in the image of the left part, we start to observe that the profile start to change, because in the image we start to change. We try to impress the viewers, saying that this microscope is like a blind man which have this stick, and they try to observe with the stick what happened or what is next in his path. This is the same with the AFM. The AFM start to scanning with the tip. The tip start to make a deflection depending of the contact of that they have, and this deflection is recorded with the changes in the photomultiplicator using the laser. So we translate a mechanical signal using the laser signal to an image. In this part, you can observe that our topography are holes in which we have like a pool, a very small pool. These pools are around 5 micrometers, but we can observe less than that in the nanonewtons, in the nano scale, because we are already using like an image size of 20 micrometers. This is a zoom of the cantilever holder, and you can observe how the laser and the photomultiplicator start to change in the detector, because they are changing the deflection of the cantilever. And as you can see in the middle part, we observe the laser and also the light for the microscope to observe the sample in real time. After a few minutes, we can achieve a very good image of our sample, and also an image with very good characteristics in topography, roughness, and also we can measurement in the nanoscale. For this part, this was only a sample, a commercial sample, but we can observe anything we want depending of the adjustment of the equipment. We can observe also in air, that is our case right now, or we can use also in water or in fluids. This equipment is very gentle because we can change the parameters and change different stuff and obtain different information for our sample. For example, this microscope also can be used for nanoindentation to know the Jones model of the samples. So this is very important because this is a very small mechanism in which we can obtain a large information for different samples. As you can see, this image is already done. We have the different patterns in our image. So after this, the image is already stored in a folder of the computer, and we can directly change for the other software that is called Nanoscope. That is the software that we use to edit and to obtain different information for the sample. After a few seconds, the nanoscope starts. We change, or we took the sample, the image of the sample that we used before, and in this part of the video I only show that this is the main power of the AFM. This is the CPU, but this is the controller of the AFM. In the nanoscope analysis, so we open this software, and this software is very gentle also, and with few clicks we can obtain very good information for our samples. In this particular case, we only open the sample, and in this particular image we obtain two different images. The first one is a high image. That means that the scale bar is made in distances, and also a deflection image that is not discussed in this part. For the first, we have to make a flatten, because the samples always try to have some irregularities. So with the flatten, we are sure that all the sample is completely flat. After that, we can use a tool that is called sectioning or section, in which we can make a line in our image, and with this line the profile is transduced to measure what is inside of our sample. So in the sectioning is used, for example, to measure fibers, to measure nanoparticles, to measure not only the diameter but also the deepness of the sample. For example, in this case we make the line, and as you can see, you can measure only a small part of the sample. In this case, this is this pool, this hole that we have in the sample. Particularly, this hole is around 5 micrometers in distance, in horizontal distance, but in vertical distance they have 46 nanometers. So we can obtain different parameters only for one image that we take in less than 5 minutes. Also using other tools that are in the program, for example the the tool that is called rudeness, we can see the rudeness of the material. For example, if we are trying to make a comparison of a rudeness of the deposition of different compounds, or the flatness of film or something, we can make a measure of the rudeness using these two different parameters that are called RQ and RA and R max. Different information can be obtained for the roughness of the material, as you can see here. In this case, this material have a very high roughness because they have holes in all the spectrum that all the image that we are seeing in the AFN. So this microscope is very gentle with all the mechanisms to obtain different images from different materials. And in this opportunity, I want to show you one part of the investigation that is already made it. In this case, we use garlic skin and garlic cellulose and agave fibers and agave cellulose. And as you can see, the high image is a topography image in which we can observe differences from the samples, and also the 3D deflection can be used to observe more detail, which sample is more flattened, which sample is more compact, or which sample we have a more nanoscale material too. And finally, we are talking about the super resolution and confocal microscopy that we have in our laboratory. This microscope is one of a kind of this kind of microscopes that we have in Mexico, and it's a glad to us to have it in our laboratory. This is a normal confocal microscopy adapted with a module for super resolution that I explained before. The advantage of these techniques of the super resolution microscopy, more than the confocal, are the see different characteristics of image in the nano scale. Also, the image have more resolution and quality to be published and to be presented in different investigations. This is the laser scanning microscopy 180. That was a project that was given for CONACYT, and as you can see, this is different from the light microscopy, because the light microscopy is a vertical and this is an inverted. This is the module of the magic happens, because they focus all the lasers on all the lamps. These are the connections from the confocal, in which you can see the gray lines are the lasers. They have 4 lasers typical, and 2 lines that common came with the confocal, that are the argon and the helium neon. These are a Schiller, only for see some parts of the microscope. You can see this part is the modulus from the incubation. As you can see, this is the heating that is connected with the incubation chamber that is in the main part of the microscope. Different with the confocals in other times, this incubation chamber is avoid the light. You can see in the top the halogenal lamp, and also the transmitted modulo to see the samples. And here are the main part of the super resolution. This is the Lyra PS1. This is the modulus for the super resolution microscope. In this module are connected different lasers, exactly 4, for different absorption and emission in the super resolution. Finally, we have this module control here in the table, also the switches for on and off, and the computer for image processing and image obtention. This microscope is a powerful tool to observe not only biological materials. First of all, we have to turn on the microscope with the main switch. After that, we have to follow the system PC initiation, and finally the components. This is the sequence, because if not, the microscope make an error. Finally, we open the SEMBlack. This is an image that I found. This is the software that we use to obtaining the confocal images. The SEN software start, and also they make a check of the system to see if everything is connected and everything is working already. This is a very intelligent machine with a very powerful PC, in which we avoid to have problems in the initiation of the samples, in the initiation of the PC, in the initiation of the confocal and super resolution microscopy. Also, because of this, the lasers are so powerful, the incubation chamber also avoid that we have several security damages. After that, we choose the locase part, in which we turn on the halogen lamp, and we can see the sample in the incubation chamber as it was a normal microscope. We have to choose which objective we want to use, and as you can see, we have 5, 10, 20, and 63 oil and water. After that, we open the incubation chamber, and as you can see, each door have a security part to avoid that these parts are open to our eyes or our individuals inside of the laboratory. The superior part of the microscope can be movable, can be up and down, depending on what you wanna do inside of the incubation chamber. As I said before, we have the transmitted light modulus and also the halogen lamp. And as you can see, this part of the microscope can be turned up to have more mobility in the incubation chamber. After that, we have to, in this case, as we use super resolution, we have to put a drop of immersion oil inside in the top of the objective that we want to use to create a meniscum between the sample and the objective and obtain a better resolution. After that, we put the sample, and we have to be careful to drop down a little bit the objective to secure very good our sample. Normally the samples are pre-prepared. We use different dyes and fluorochromes and different techniques to obtain very good samples. Finally, we have to close everything, and if in this part we make a mistake, we forget to close something, or we don't put the cabezal in the very good position, the software send us an error to check some part of the microscope. Normally, each part of the microscope have to have one or two security paths, so we can check twice everything. The same mechanism that the vertical microscope, we can see the sample by binoculars. We can move the sample using a joystick that we have in the right part of the of the microscope, but normally the sample can be seen from this part. So now in the software we turn off the light and we choose the acquisition mode. In acquisition mode we have to choose the different obtentions. In this case, laser scanning microscopy and channels. We also can use lambda mode or only online fingerprinting. For this image that I wanna see, we are using the smart setup, and we choose DAPI as the first fluorochrome. Also, Alexa Fluor 48080 and DS Red. This in this part of the software we can choose if you wanna the fastest signal, the best signal, or the smartest signal. After that, the software choose the better characteristics to obtain the image, and we only can change the resolution of the image. We can change also the velocity in which we want to take the image, and finally the laser gain. As you can see, the image that we obtained for this sample is very good. We have 3 different fluorochromes for 3 different parts of the sample, and after a best fit we can observe how the 3 different parts are dyed in a different color with a different composition of the fluorochrome. This image is very good in comparison with other images that we want to have, but this is a sample, a commercial sample. After that, we can choose another techniques. In this case, we want to show you SIM, that is a structure illumination microscopy, and this is a super resolution technique. After choose the mechanism, we have to make the same that in the confocal, but in this case we only talk about lasers. We don't talk about fluorochromes, so we don't know if a fluorochrome can emit another emission line. For that reason, we have to choose the perfect bandpass filter to the adequate fluorochrome. So after that, we can see the image for the structure illumination, and as I show you in this part of the video, we have different rotations. That is the main part of the main importance of the structure illumination microscopy, that the image are taken with a grid, and the grid is moving in 3 different angles, so in which in different angles we obtain different composition of the image. As you can see in the top part of the video, the grids are making a composition of the final image that we have. So after best fit of this image, we are able to processing this image to the other part of the analysis. We choose processing tab, then structure illumination, and then we choose the analysis of the structure illumination. We select the image and we make the analysis, making apply. In instance, for this image, the software start to edit and to compilate each fluorophore with each emission and with each emission wavelength. So they compile each color and they put together in a final image. We are able to make in some adjustments inside of the sample, because we know that the metadata that is obtained is big, so we wanna make an image that is better For example, we can choose one single channel and start to modulate the absorption or the emission depending of the color that we want to see. In this case, we reduce the blue and we only left the mean part, and also with the red we erase everything in the background and we left only the part that really emit the red color and the green color. As you can see, the details even with a camera are better than in a confocal. Normally these kind of techniques permit or allow us to obtain more detailed information for a sample, and also the interaction between a sample and a processing or a treatment that we make in the sample. To make a better comparison, we took another software that is called SemBlue, and we compare the first image that we took with the confocal and the second image that we took from the SIM. And as you can see, the detail is good in red but not in green, in comparison with the SIM image, in which green and red are good. The definition of the fluorochromes and also of the image and the pixels of the image are better using the structure illumination microscopy. So in comparison, both techniques are great, but we know that with SIM we can have better resolution in the nanometric scale that we don't obtain in confocal.